We are very fortunate today to have our second speaker in 2 weeks who is born and raised in New Jersey. So this is really wonderful that we're getting a lot of representation from across the US, particularly one little area. All kidding aside, it's really my pleasure to introduce today our speaker, Dr. Kristen Brown, who's a research fellow at the National Institutes of Health, specifically the National Human Genomic Research Institute, NHGRI, where she does a lot of research trying to, again, I think this is a theme that many of you will see throughout the series, trying to bring together sort of some disparate fields to draw some interesting interdisciplinary insights into the social determinants of health. I'm particularly trying to understand mechanism and looking at some of the, I can read your subtitle, genetic embodiment of social determinants, which I think is really wonderful, these biological embedding of your social environments. Dr. Brown did her undergraduate in the great state of New Jersey at Rutgers University, then went and did her master's and PhD at the University of Michigan in Ann Arbor. After that, she went to NHGRI, where she is today. She's got many fantastic publications, some of which she shared with us in advance of this seminar. And she's won many awards, one of which at least is based on her 2020 publication in Psychoneuroendocrinology, looking at the social regulation of inflammation-related gene expression in the multi-ethnic study of atherosclerosis, the MESA study that I think she might talk to us a little bit about today. And I promised I would keep it short and sweet and glowing. So without further ado, everyone, please join me in welcoming Dr. Brown.
Okay. Hello, everybody. Thank you so much for that introduction. It is my pleasure to be here with you all today, presenting some work which I've entitled A Multi-Omic Approach to Precision Medicine, Genetic Embodiment of Social Determinants. Just the standard NIH disclaimer. This presentation was prepared by me in my personal capacity. The opinions expressed in this article are my own and do not reflect the view of the NIH, Department of Health and Human Services, or of the United States government. Let me also give you a little bit of fair warning that I live across the street from a fire station. So if you suddenly hear fire alarms, I promise you I'm okay. Normally I don't notice it, but everybody I talk to does. And apparently there likes to be emergencies when I'm on Zoom calls. So just, yeah, I'll be okay. So about me, I like starting here because, especially when I'm talking to students, because I don't think the journey was so direct or so intentional, for lack of a better word. But I am from the great state of New Jersey, from a town called Neptune. I used to say that I was from the Jersey Shore, but for anybody who's actually watched that show, I don't feel like I can say Jersey Shore. Oh, there goes the fire truck I was talking about. But for anybody who, I knew it was coming. But for anybody who's seen the show Jersey Shore, after that show came out, I don't like saying I'm from the Jersey Shore, but I am indeed from the Jersey Shore. I stayed in New Jersey to go to college. I went to Rutgers. And at Rutgers, I double majored in biology and psychology. So I knew I really liked the biomedical sciences. I like knowing how the body works and how the heart changes when you're exercising and how the lungs react and those type of things. But at the same time, I also like psychology, what made people behave the way that they did. I also minored in religion. So the story that I like to say is that I like knowing how people function, mind, body, and spirit. The thing is that I remember being a senior and saying like, okay, what job do I do with this? I know I like knowing how the body works. I like knowing how people behave. At the time, I thought I had to pick one. Do I want to be in biomedical sciences or do I want to be a psychologist? So I did what any self-respecting 21-year-old would do, and I stalled. I stalled for a year. And I actually did a post-bac program at the University of Michigan, working in a research lab in physiology, cutting up mice, doing cell cultures, doing PCR, all that type of work. But the point of the post-bac was to go to graduate school. I said, okay, well, what am I going to go to graduate school for? Again, I was faced with this question. Do I want to be a biomedical? So I was like, okay, well, Kristen, you have to pick. So that's what I did. I actually started my PhD. This is something that doesn't show up on my CV, but I started my PhD in the Department of Molecular and Integrative Physiology at University of Michigan. again, cutting up mice, looking at different protein expressions, doing Western blots, that type of thing. And I got a couple months in and kind of had a heart-to-heart with myself and realized that I didn't like it. And fortunately for me is that Michigan has what's known as an umbrella program. So you spend your first year just deciding which program you're going to be in. Do you want to be in physiology, genetics, cell biology, for example? And you rotate through different labs, trying out these different paths that you can go. Me being, I guess, a little bit of a rebel to the system, as I was looking for which lab I would go to, I came across this website. And the website was called the Center for Integrative Approaches to Health Disparities. And this center, it happened to be at Michigan, over in a all the way across campus in the School of Public Health. And it was run by a social epidemiologist named Ana Diaz-Roche. She is an expert on understanding how the neighborhood environment affects your health. And also part of that center was a genetic epidemiologist named Sharon Cardia. And I was said, wait a minute, how is a person who studies neighborhoods and neighborhood level stressors working with a person that studies genetics? How do you integrate it? So I set up meetings with both of them and realized this is what I wanted to do. This is what I've been looking for, was an opportunity to integrate being a person that studies social sciences and genetic sciences at the same time. So the work that I'm going to present today, so you guys get to hear about it in a little bit. This is the path of how I got there. After I left Michigan, I briefly postdoced at Emory before being recruited for a position at NHGRI. And specifically, I'm in the social and behavioral research branch in the social epidemiology unit. So again, I'm in a position that allows me to be both a genetic epidemiologist and social epidemiologist. If I'm going to talk about the focus of my work, it's really to answer this one question. How do social determinants of health and the genome together affect disease risk? And how does this relationship matter for racial health inequalities? So social determinants of health. We know there's longstanding, well-documented research knowing that social factors are important for health. Socioeconomic status is considered a fundamental cause of health in the sense that having access to resources, your level of education, where you're able to live, for example, that those factors contribute to your health outcomes. Similarly, we know neighborhood environment. You live in a neighborhood that is walkable, where you can walk to stores and get access to your bike, for example. Psychosocial stress, things like discrimination, loneliness, we know that those matter for health, as well as access to quantity and quality of healthcare. Can I afford to go get a physical every year? Can I engage with the healthcare system in a way that I can get access to preventative services and be treated with equal respect when I'm there? But at the same time, we know that genes matter too. And not just for Mendelian diseases. So Mendelian diseases are diseases where it's pretty much one gene that affects your risk of getting that disease. So for example, like sickle cell or Tay-Sachs, but also for complex diseases. So complex diseases are the diseases where it's multiple genes that affect the risk of you getting that disease. So how do we think about the relationship between the environment and genes when we try to figure out health outcomes? Well, there's a few ways. One is the longstanding nature versus nurture debate. I remember learning about this way back in elementary school. And with the nature versus nurture debate, you're trying to figure out what component of your probability of getting a disease, diabetes, for example, is due to your genetic predisposition, and to what extent is it nurture? What extent is some of your experiences? So one of the ways in which we try to get to this is using twin studies. So for example, let me know if I start going too fast. I tend to do that. But we use twin studies. So in a twin study, what you do is you compare monozygotic, those are identical twins, versus dizygotic or fraternal twins, and try to use those relationships to tease apart what part is genetic and what part is environmental, with the assumption that monozygotic twins have the exact same DNA, in pretty much the same environment, whereas dizygotic twins have the genetic similarities of siblings. So they have about 50% of their same DNA in pretty much the same environment, and try to teach and use that difference to try to figure out what's genetic, what's nature, and what's nurture in a phenotype. So here I'm just showing a graphic from looking at systolic blood pressure, the coordinates between systolic blood pressure for twins. And you can see that the monozygotic twins, identical twins, have much higher correlation of their blood pressures compared to the dizygotic twins. And that's how you would try to tease apart the nature versus nurture debate for systolic blood pressure. Another way that we think about the relationship between genes and health is looking at gene by environment interactions. So given that you have a certain genetic profile, how does that genetic profile perform in different environments? One of my favorite examples of that is the idea of perfect pitch. So perfect pitch idea is that people currently think that you genetically have the machinery, quote unquote, to have perfect pitch, but you have to be musically trained as a child in order to ever get it. So you can have the genes, but if you're not trained, then you won't have perfect pitch. You have to have both. So I like this quote by Charles Brewer, he's a psychologist, who says that heredity deals the card. More recently, we started to think about this different way of looking at this environment and genes relationship in a field called human social genomics, which has probably existed for about a decade, a little bit over a decade now. And the idea of human social genomics, or at least the way that I like to look at it, is that you're trying to see how does the social environment affect how your genes are functioning. So whereas the other two ways of looking at genes and environment, nature versus nurture, and gene by environment, it was kind of like nature and, it was genes and environment on one side and on the other. And then, but with human social genomics, you're trying to really get to how does the social environment affect how the genes are functioning. So this quote by George Slavich, who's one of the originators, demonstrating that the external social conditions, and especially our subjective perceptions of those conditions, can influence our most basic internal biological processes, namely the expression of our genes. So one of the early studies in this field was by a guy named Steve Cole at UCLA, who writes this 2007 paper called Social Regulation of Gene Expression in Human Leukocytes. And what he wanted to do was to determine if he looked at loneliness was his exposure of interest. And he said, is loneliness associated with gene expression? And if so, which genes are they associated with? So it was a relatively small sample size that he had. He had 14 people, 8 people that said that they were low lonely individuals. He took the extremes of his bigger data set. And he had 8 people that were low lonely individuals and 6 people that were high lonely individuals, and tried to compare across the genome, are there differences in gene expression by loneliness status? And what he found was that there were 144 genes corresponding to 209 gene transcripts. That difference is because of alternative splicing for those in the genetics crowd, that were differentially expressed between the loneliness groups. And he considered differentially expressed to be a 30% difference in expression. So then he had this long list of genes. He said, okay, well, is there a biological story associated with these differences? So he used gene ontology, which is a bioinformatic database. And what it pointed to was an overrepresentation of genes involved in the immune response. So essentially what this study showed is that loneliness was affecting the genes involved in your immune response. Really, really cool study. Really, really cool study. He was definitely an inspiration for this entire project. So what happens when somebody has a really, really cool study is that other people try to do it with their favorite exposure. So over the years, other scientists then said, like, okay, well, this worked for loneliness. Will this work socioeconomic status? What do we see if we look at grief? What do we see if we look at caregiving stress? For these different adverse social exposures, that there was a pretty consistent pattern, which they then called the conserved transcriptional response to adversity, or CTRA. And the CTRA was characterized by an upregulation of pro-inflammatory genes, a downregulation of type 1 interferon response genes, and a downregulation of antibody synthesis genes. So essentially, that being exposed to an adverse social experience was associated with the suppression of your immune system. Great work, but there were some important limitations that I thought were noteworthy. One is that most of the studies had pretty small sample sizes. Two, they had varying thresholds for differential expression. So some studies considered 30%, another study said, is there a 20% difference? Another study said, is there a 50% difference? So it's really hard to make these cross-study comparisons when they're using different cutoffs for what they're considering differential expression. Third, there was no consensus on most appropriate method. So people use different analytical strategies to try to get to this question, which would also affect getting different results. Fourth is that studies were conducted in differing cell types, specifically what are known as peripheral blood mononuclear cells. Peripheral blood, PBMCs, are essentially a hodgepodge of different white blood cells, which becomes an issue because it's suspected that gene expression is cell type specific. So the expression patterns that you may get in monocytes may be different from what you get in macrophages or B cells. But if you put all these cell types together, you may miss something that was actually there. And last, there hasn't really been replication, which leads me to project one that I'm going to talk about today. So the goal of my first project was to conduct a large epidemiologic study assessing the association between a range of social environmental factors and expression of the set of genes previously implicated as part of this conserved transcriptional response to adversity. So essentially, I wanted to look at a bunch of different social environmental factors. I list them here, but I'll talk about them more in a bit, and see if they were associated with the same genes that other studies found an association with. So I used the MESA data set. MESA is a longitudinal study designed to investigate subclinical markers of cardiovascular disease. There's been, actually, I believe they've very close, if not done, with exam 6 now, but they started in 2000. And they conduct this study every 2 to 3 years. The participants were age 45 to 84 at baseline. And what makes MESA such a great data set to ask this question is because they have rich data collected on both the social and psychosocial exposures, but also have genetic data. So typically studies that had good genetic data had very, very basic social information. But in MESA specifically, you have good genetic data, but you also have a very good range of social and psychosocial exposures. Specifically for this work, I used a subset of the MESA data set. They only had genetic data in exam 5. So at exam 5, which was collected from 2010 to 2012, it had 1,264 participants. This is the racial ethnic breakdown, as well as the sex breakdown, and the study sites of where they came from. So I looked at 6 different social environmental factors. First, I looked at socioeconomic status. So I looked at it both in adult SES and let me also say this, I don't mind if you interrupt me. I know that my work tends to be a bit interdisciplinary. So if you have a burning question or something, feel free to interrupt me. So I looked at socioeconomic status. It was collected in exam 1. It dichotomized it as to having a college degree or higher. And I looked also at childhood socioeconomic status, which I considered a parent who had a high school diploma or higher. Next, I used the perceived stress using Cohen's perceived stress scale. This question asks, in the last month, how often have you felt unable to control the important things, confident about your ability to handle personal problems, things were going your way, or difficulties were piling so high that you could not overcome them. Participants answered this question via Likert scale. So that's one of the survey questions where it says strongly agree, neutral, disagree, strongly disagree, for example. This loneliness, I use loneliness, and I do want to note that this is the same loneliness scale that was used in the Steve Cole study that I mentioned earlier. And this question asks, how often do you feel that you lack companionship, left out, isolated from others? Major lifetime discrimination says, have you ever been unfairly fired or denied a promotion, not hired for a job, unfairly treated by the police, unfairly discouraged from continuing education, prevented from moving into a neighborhood, or neighbors made it difficult for you? Chronic burden, it gets to, as it suggests, a more chronic stress measure. And it said, do you have serious ongoing health problems for yourself, serious ongoing health problems for someone close to you, ongoing difficulties with your job or ability to work, ongoing financial strain, ongoing difficulties in a relationship with someone close to you? And I coded this as that the ongoing problem, because I was trying to get to a more chronic measure, it had to be at least 6 months, and the participant had to say that it was moderately or very stressful. So if this person had a health problem, but it had been less than 6 months, and they said, yeah, I'm kind of sick, but it's not stressing me out, then it was not considered. And then I looked at social support, which was measured in exam 5. And it says, how much can you rely on friends and family to help? How much can you open up to them? How often do people make too many demands on you? Or do people like, hold on 1 second. So I'm choking on my water. Or do people let you down when you're counting them? So then I had to figure out, okay, what genes am I going to include in this study? So I did a Google search, or a search of Google Scholar and PubMed. Give me 1 more second. Sorry about that. And I looked up this term, conserved transcriptional response to adversity, because I wanted to be able to catch all of the genes that had been found in the previous study to be, in previous studies, to be associated with gene expression. And when I did that, it gave me 63 studies. I excluded, my inclusion criteria included only primary studies. It had to have gene expression as an outcome. So for example, I excluded literature reviews and things like that, or things where gene expression, it was just mentioned in the intro or something like that. It had to be at least in primates. So I didn't include like mice studies or anything like that. And when I did that, it gave me a total of 8 studies. And I also added the 2007 loneliness paper that I talked about earlier, because although the phrase wasn't used, it wasn't used because it pretty much wasn't really invented yet, but it was the seminal paper of this work, which gave me a total of 9 studies. So this is the list of 9 studies. You see the 2007 loneliness paper by Steve Cole, a range of exposures, caregiving stress, rearing. This was actually in monkeys. So it was like, was the monkey reared by their mom versus a surrogate of some sort? Well-being, socioeconomic status, variety of different exposures that people had looked at. And in column 3, you see the number of differentially expressed genes by those each exposures. And in the 4th column, the threshold for differential expression. So that's what I was talking about earlier, where at the time I was doing this work, there wasn't really a consensus on like, okay, this is where the cutoff should be. So there was a big range. And then in the last column, you see the sample size. So earlier when I said that there weren't really big sample sizes, this is the sample sizes of each study. So using MESA was very beneficial because I had 1,200 people, 1,264 people, which by far had exceeded the sample size of the previous studies. So in trying to figure out what genes am I going to consider to be part of this transcriptional response to adversity, I summed up all of the genes across these different studies. And when I did that, there was a total of 1,305 unique genes, which corresponded to 1,854 gene transcripts. So for an approach, as I also mentioned earlier, there was really no gold standard on how to do this. So I applied 3 distinct but complementary approaches. First, I used a study known as global ANCOVA. So global ANCOVA is what is known as a self-contained gene set enrichment analysis. Essentially what it says is, okay, I want to know, are any of these genes associated with my exposure? So the benefit of doing that is that it lessens your concern about multiple testing. So it gives you one question, it gives you one p-value. Is loneliness associated with any of these 1,800 gene transcripts? Yes, no. The downside of it is that it doesn't identify which gene transcript is associated with the exposure. So it's a good first pass, but all it is the first pass. Next, I use linear regression. Linear regression has its issues in this type of work, so linear regression, in contrast to the global ANCOVA, it's a one-by-one analysis. Is loneliness associated with gene 1? Is loneliness associated with gene 2? Is all the way down to 854. I use it because it's familiar. People know what it is. It's easy to interpret. It's easy to say what beta is or means, but it doesn't work well when the number of genes is greater than the number of people. So to kind of fix that, I used a penalized regression technique known as ElasticNet. And ElasticNet was developed to help improve prediction and interpretation above the linear regression type model. It's a hybrid of what's known as ridge regression and lasso. So essentially what it does is it pulls out, okay, so if I want to know, is loneliness associated with these 1,854 genes? If it pulls out, if it does variable selection, so if gene A is associated with loneliness, it will also give me the genes that are highly correlated with gene A. So if gene A is highly correlated with gene M, it'll say, okay, A and M are both associated with loneliness. It works well when the number of genes is greater than the number of people, which is the situation that I had here, where I had 1,854 gene transcripts, but 1,264 people. The problem with it is that it's difficult to interpret because it does the shrinkage. You don't really know what that beta coefficient means anymore. So these are the results of first of the global ANCOVA test. So these are the different variables that I assessed. And I found significant associations for discrimination and chronic burden, and marginally significant associations for loneliness and adult socioeconomic status. When I look at the linear regression results in this column, you can see the number of just going at p equals p is less than 0.05. You can see the number of transcripts that were selected or that were less than p equals 0.05. But I do want to note that none of the transcripts were significant after false discovery rate multiple correction. And false discovery rate is a little bit of a liberal correction to even use. So what this kind of shows or starts to show is our standard technique of just doing a knee-jerk linear regression was not going to be sufficient for this genomics type work in which we have highly correlated variables and a lot of hypotheses. So then these are the results from the ElasticNet. You can see, and what I do want to notice is that even between the ElasticNet and linear regression results, that not perfectly, but somewhat of a pattern is that the the exposures that were significant in GLOBA and COVA, there tended to be more somewhat significant results in the linear regression and ElasticNet. So it kind of hints to it, although it may not be statistically over the line to say. And then in the last column, I'm trying to show that the overlap, there was significant overlap between the linear regression and ElasticNet results. So even if ElasticNet, the genes that ElasticNet pulled out, it tended to be less than 0.05 in the linear regression analysis. So then I was really interested in this, the overlap again between the exposures. So, okay, so with these genes that I'm finding to be looking somewhat significant, are they the same genes that are being picked up across exposures? And for the most part, they weren't. There were a few that overlapped between at least two of the variables as shown here. But so I said, okay, is there a biological story, like, or is there a certain pathway that I can pick up on that if a gene is associated with both discrimination and loneliness, for example? And not really. So when I looked at the list of genes, some were involved in immune response, an interferon gene, interleukin, but others were not. Things in organelle development and axon guidance. And so there wasn't really a biological story to really make there. So then the question is like, okay, well, how does this fit into what we already knew? So in the loneliness paper, again, that I consider the seminal paper of this work, Steve Cole found 144 differentially expressed genes. And because of the way I designed the study, all 144 of those differentially expressed genes were in this analysis that I'm looking at now. But only 6 of them were selected via ElasticNet. So that's really small overlap. I was a little bit surprised by that. The other exposure that also overlapped with the previous study was adult socioeconomic status by Powell, and Powell found that there were 384 differentially expressed genes. But in my study, only 4 of them were selected via ElasticNet. So these dissimilarities were likely due to differences potentially in the methodological approach between the previous and present studies, or possibly just having a different sample. So what's the take home, I guess, from this project one, is that it provides some evidence that there may be a concerted conserved response at the genome level based on the global analysis and ElasticNet. But, and a big but, I would say at that, is unclear of what the characteristics of exposures that suggest significance. So it's curious to say that, okay, adult socioeconomic status was marginally associated, but childhood socioeconomic status wasn't, or that we picked up on something with discrimination. So I guess one of the questions that it's one of those things where trying to answer one question gave you more questions. So, okay, well, are there characteristics of the exposure that are more likely to be significant? So for example, is it ongoing stressors that may be more important than having one single event? Or is it one single event, but it was very impactful as to where I might be able to pick up a transcriptome response? And replication of individual genes is difficult across studies. So it may just be better that instead of trying to replicate individual gene findings, that maybe it's better to go more of a concerted pathway analysis. Which brings me to project 2. So project 2 is very, very related to project 1. It's just really with a different set of genes. And I wanted to know, this is the paper that I had you guys read, but I wanted to look at these range of social environmental factors and say like, okay, is this associated with inflammation of genes that we know are involved in inflammation specifically? Why look at inflammation? So here I list the leading causes of death. Obviously this has changed now with COVID being on these leading causes of death. But on this list, most of these diseases have some sort of, or there's evidence that they have some sort of inflammation component to it. When I say inflammation, I'm not meaning like, you know, the acute inflammation when you cut yourself, like the inflammation when a low-grade chronic inflammation that is ongoing. So for example, there's evidence that high levels of CRP, that C-reactive protein, which is used often to measure inflammation, gives you a 1.6 times the risk of ischemic heart disease. Evidence in diabetes where IL-6, NCRP, interleukin-6 is also a measure of inflammation, increases risk of diabetes, and similar with stroke. Again, for this project, I used data from MESA, which I talked about earlier, and the same statistical approach. This time I only did the linear regression and elastic net if it was significant in global and COVA, because the rationale was that global and COVA is supposed to tell me, are any of these genes associated? So I only followed up using for linear regression elastic net when it was significant. So how do I decide which genes I'm going to look at as being inflammation genes? I used a bioinformatic database called Genontology, and I looked at 4 different biological processes: chronic inflammation, inflammatory response, regulation of the inflammatory response, and the immune response. Here I'm showing the overlap in the genes between those 4 biological processes. As you would imagine, that genes that are considered part of chronic inflammation would also be considered part of the inflammatory response. So this chart is just showing the overlap between the genes. And okay, so here I'm showing the global ANCOVA results first for chronic inflammation. So you can see that global ANCOVA is saying that loneliness, chronic burden, and discrimination are associated with chronic inflammation as a biological process. But again, global ANCOVA doesn't tell you which of those genes it is. When we look greater at the inflammatory response, we see discrimination associated with the inflammatory response. Again, discrimination in the immune response. And lastly, discrimination with regulation of the inflammatory response. And then when we look at the findings for the linear regression and elastic net, we were able to pick up, using linear regression, some genes that are likely to be somewhat influenced by the exposure. But again, as in project 1, none of the transcripts were significant after false discovery rate. But then again, we use elastic net, and then we were able to pick up on some of the genes within each biological process to be associated with the exposure. And as again, in project 1, we did see a good amount of overlap. So if the genes were selected via elastic net, they tended to have lower p-values in the linear regression. So what does this study show? It demonstrates that some social environmental factors are associated with inflammation at a system level. And among the social environmental exposure, discrimination may be especially influential in affecting inflammatory gene expression. Strengths and limitations. I think one of the strengths was being able to use the MESA data set, that it's a large racially ethnic diverse sample at 1,264, which exceeded that of most of the other studies. It has, again, the collection of social and psychosocial exposures, as well as gene expression collected solely in monocytes. So it gets you around that PBMC issue. And also the use of varying methodologies that account for the complex nature of gene expression. So I was able to use, especially using the elastic net, which allowed me to do variable selection and control for the correlation at the same time. Limitations. Well, one of the limitations is that the exposure and outcome variables were collected at different time points. So gene expression in MESA was collected in exam 5, whereas the exposures for the most part were throughout at that time, up to 5 MESA exams. So for example, socioeconomic status was captured in exam 1. I believe chronic burden was like in exam 4. And the problem with that is that we don't know how dynamic gene expression is. So if we collected your your stress level at one time point and then took your gene expression profile 10 years later, it's hard to really say for sure that those things were associated. What I did try to do to minimize that is I always tried to use the measures that it either one didn't matter or two was closest to exam 5 when gene expression was collected. So for example, I did socioeconomic status and I used the measure of education. Well, if you had a PhD at exam 1, hopefully you still had that PhD at exam 5. Education typically does not get taken away from you, I guess, unless you do something really, really terrible. Second is that the findings need to be replicated in a separate epidemiologic cohort. The point of really Project One was to try to be somewhat of a replication for the prior studies, but even this, and even as great as MESA is, replication is always needed. And third, gene ontology itself is not perfect. So when I was trying to get to, these are the inflammatory genes, gene ontology was just a great way to do it, but it's not perfect. We don't know all of the inflammatory genes yet. Future directions. I think it's always a good thing to know where you want to go next. And I would say that it is a great time to be in science. And this is why. The idea of precision medicine really, really excites me. So this is a quote from Francis Scott Collins, director of NIH in 2015, where he was unveiling this idea of precision medicine. And he said that the initiative will encourage and support the next generation of scientists to develop creative new approaches for detecting, measuring, and analyzing a wide range of biomedical information, including the molecular, genomic, cellular, clinical, behavioral, physiological, and environmental parameters. So it's really, science is moving in a way to be interdisciplinary and to try to see how these different aspects work together in order to try to create more precise prevention and treatment approaches. What are the things that I think we need to do? On the social side, I think we need more comprehensive measures of the social environment. So if I'm filling out a survey right now, I'm not just my loneliness score, right? Like Kristen Brown has some level of loneliness. I also have some level of education, some level of work stress. And I'm not any one of those scores and one of those scores by itself. I'm some better complete picture of a social environment. Also, a lot of this work so far has done mostly adverse exposure. What's your stress level? How low is your socioeconomic status? How lonely are you? And I think that, and I don't know why scientists just do this anyway, but we need more effort on like not being from a lack standpoint, but being from a better standpoint. So I think we need more protective studies to say like, okay, well, having a mental health therapist is important, or, like those type of things that are like, what is good for you, as opposed to always thinking from a position of lack. Next, I think we need further exploration of the biological mechanisms through which social factors act to affect health. So what is regulating gene expression? So obviously I'm thinking in the epigenomics world now to say like, okay, not just to look at gene expression, but also let's look at sequence level, let's look at the epigenome, let's see how those things all go together. But also, once we also have gene expression, where does it go from there? So then how do we link gene expression then with proteomics and metabolomics and greater physiological processes? And of course, we always need longitudinal studies, obviously to avoid issues of reverse causality. I always come from the standpoint that it's your environment affecting your genes. There are other people who think in the opposite way, that genes are affecting your environment, which can get, I think, and that's one of those ways that I think in current genetics is, I don't know how to put it. It's a little scary to me, because there are some scientists who come from the perspective like, okay, I can look at your genes and determine your level of education, or I can look at your genes and determine whether you're going to end up in jail. And I always come from the social, on the genomics or the social integrating with the genomics to try to do my work. But I think we need those longitudinal studies to be able to assess directionality. And also to understand how these factors matter differently across the life course. So if I face discrimination as an 18-year-old, is that different than as a 50-year-old? Or if I'm lonely and I'm 20, is that different than being lonely and 75? In other words, I think that the future of human social genomics is promising, and there's a lot to be done. I really enjoy the field. I hope I've convinced you to as well. Just want to acknowledge my key collaborators. Funding is always a great thing, and obviously the participants, Lisa. And with that, I'll take any questions that you guys have for me.
Thank you so much. That was fantastic. I think your future directions ended on a really fantastic note, which is that environment trumps genes 10 times out of 10, which doesn't mean genes aren't meaningful. It just means that they're a nice message that there's a lot we can do outside of, the genetic effects to help improve conditions of everyone, health of everyone. So I'm going to open it up to questions. Does anyone have any questions for Dr. Brown? Ben Trumbull has a question. Do you want me to read that, or do you want to ask it, Ben?
Sure, I can go ahead and ask. Thanks so much for a great talk. I was just wondering, did you see any interactions between, say, like discrimination and loneliness, or discrimination and other variables?
Great question. I actually have not looked yet. I'm more so thinking in like, how do we comprehensively pull out the social factors that may matter? So I think that's more of the direction that I'm thinking of going in. But yeah, I certainly would expect that there is an interaction effect if I looked closer at these variables. It makes sense, right?
Yeah, no, cool stuff.
Thank you.
Other questions? You can raise your hand. All right, well, you can raise your hand on video. Sylvie, you're up.
I'm trying to switch it up. My blue hands or not? Just that more like a very general question, I guess, about where this field would be going. So imagine, like 10 years from now or 5 years or next year, you have like the genes that are correlated with some adverse effects. Like what's the next step with that? Like once we've isolated those particular genes that are involved, like what can we do with that? Like, are there some interventions that you imagine, or is it more about, we understand more these like gene environment interactions?
Great question. I would hope that it would set the stage for prevention and treatment, right? Of saying like, okay, if I know that me being lonely is increasing my risk of heart disease via an attack on my ability for my immune system to function, I would hope that that lays the groundwork for the people that create interventions, like create the more social interventions, but also for the clinicians, right? So I thought it was very interesting. I guess personal note, I have a new PCP I went to last month, and during her like normal, health history, I'm normally used to does your dad have heart disease? Does your mom have heart disease? But they also had questions in there about like depression. They had questions in there about loneliness. And I thought, and I told her, I was like, I may be a little bit of a nerd in this, but I'm loving that you're asking this, right? Because we know that loneliness is associated with health. And yeah, I think it is good for my clinician to ask me, do you live by yourself? Do you have people you can depend on? Because maybe how she treats me is different, that maybe I don't need depression medicine. Maybe I need, I don't know, to call my mom. Like, I don't know, like maybe how you can tease apart and treat the individual, I would hope would be better and more precise, I guess, to use NIH's word. Wait, I see something in the chat who said, I can't.
Yeah. Lexi, I'll read out your question. So to what extent does personal perception of social adversity prevail over the actual environment. So perceived versus actual, like status or adversity or loneliness, right? I know that Steve Cole and others have done some work on that. For example, two people could be discriminated against, but maybe one of them is not as bothered by it as the other. We see different health detriments or health consequences based on these different perceptions.
Great question. So the idea right now is that your subjective experience matters a lot, right? Because the way that it's believed to act is through your HPA axis, or essentially your fight or flight response. If there is a lion sitting right here in my living room next to me, and I'm not scared of lions, my body is not going to react to this lion, right? But if my body's like, oh no, lion, right? My heart's going to start increasing its heart rate. My lungs are going to start doing certain things. My reproductive system is going to like, we don't need you right now. We've got to put all of our energy in these things to get away. So the way that your body reacts to things that are considered bad for you, but you have to perceive them as bad for you, are what we know is embedded biologically. At the same time, there is something to just being in certain environments, right? So where I live, I rarely drive because I can walk. It's easier for me to walk to Trader Joe's than it is to drive to Trader Joe's and fight for parking. So being in an environment that is walkable is just inherently healthy, whether I perceive it that way or not. But I guess to answer your question, basically, is to say that yes, perception is probably a bigger part than even just the reality, but the reality is still important. But great question.
Awesome, thank you. Other questions? I'm gonna sort of use one of our students who submitted a question online ahead of time, which is, and you mentioned this, we need to do some longitudinal studies to really understand how sticky your past social environment is. So, can you tell us what we know about the reversibility of some of these negative health consequences, let's say at the gene regulatory level, in terms of if you improve someone's social situation or improve their social environment, do you see concomitant improvements in their health, or at least at the gene regulatory level? And there's always two ends to this spectrum. If someone is in a better environment but moves to a worse environment, how does that change? How sticky is their earlier environment?
From what I understand, the thought is that it is likely to be somewhat reversible. So for example, there's studies in like behavioral therapies, and they show a good positive thing. So it's like, okay, if my hypothesis is that like a lot of this is really working probably through like the epigenome and trying to get to transcriptomics. But I think we need more work to tease apart, like, okay, what is the genome doing? What part is epigenomic? Because if it's epigenomic, to some extent, we think we can revert, we would hope that we could like reverse it, as opposed to like being at the DNA sequence level, which, is relatively stable throughout your whole life. So I think that that's where the research really has to go, is to try to tease apart, okay, what level are we really impacting even when we talk about gene expression? And then how do we reverse it? And how do we just say like, oh wait, this isn't methylated anymore, so you should be happier, like, or healthier. So I think that's what we kind of like tease that apart.
Wonderful, thank you. For non-human animals, and maybe some of those things, but that's my very cool. Any other questions from the broader group? Taylor asked a question here. How does social adversity change from one generation to the next? For example, if someone with a low SES growing up becomes more educated than their parents, gets a better job, how does this affect the person's kid? You know that up to 3 generations are affected when a baby is in utero.
That is a great question. I don't think that we know yet. And I think it will depend on the exposure itself. So there has been a study showing that like, it was, I haven't read this in a few years, but people who survived the Holocaust, that they found different epigenomic signatures, and I think either their kids or grandkids, compared to those who didn't. So we do think that there may be some sort of intergenerational transmission. But as far as yourself, and I think what you're getting to here is like more of like a life course question. I know that there is some data about how your life course, right? Like I'm still me. So I grew up with 2 working parents. So I ate a lot of fast food. Now I have a whole PhD and I eat Trader Joe's, but I still probably eat more McDonald's than a lot of my colleagues, right? So there is some evidence that you are also the sum of your experiences. So I think it probably depends a little bit on the person, like, yeah, of how much your childhood still influences your adulthood, and how much it's like a combination of those things, and how much of it is like, did I just become a whole new person? I don't think you become a whole new person. Who you were as a child is still, to some extent, part of you. So, great question. Wish I had a better answer, but it's one of those future research things that I think we need to better understand.